Hello and welcome to Unit 15 of Introduction to Biology's Introduction to Anatomy and Physiology. In this lecture we're going to discuss the digestive system. That includes the alimentary canal and the associated organs, accessory glands, and this material can be found in chapter 23 in the book. I'm going to begin by discussing the alimentary canal. The alimentary canal is a tube, a long continuous tube that leads from the mouth to the anus, basically. So that includes the mouth, the pharynx, which is the hollow area behind the mouth, the esophagus, which is the tube that leads from the pharynx or the nasopharynx area to the stomach. Then we have the stomach. Then we have the small intestine. The small intestine is divided into three parts called the duodenum, the jejunum, and the ileum. Then we have a kind of an enlarged sac that the small intestine joins into called the cecum. And then we have the large intestine, which is also known often as the colon. And then the rectum, which is the whole kind of apparatus around the anus, and then the anus, so the mouth is a way for food to get into the body. The anus is a way for food to get out of the body, the waste products that we don't digest or that we don't fully digest. And that is one continuous tube. It's one continuous long tube that is referred to as the alimentary canal. Now, feeding, there are various what are known as accessory organs that kind of dump things into the alimentary canal. They're feeding things into the alimentary canal, and those include the liver. The liver doesn't actually put anything into the alimentary canal, but it is used as a filter to detoxify the food that is absorbed in the intestine. So most of the time when we break down food, digestion of starch, amylose begins in the mouth because there are amylase enzymes in saliva. So the saliva is produced by the salivary glands. And saliva contains amylose enzymes, so technically the digestion or the breakdown or the hydrolysis of amylose begins in the mouth with the amylases that are contained in the saliva. Then when the food travels to the stomach, the stomach produces a protease enzyme called pepsin that breaks down the peptide bonds that holds proteins together. So remember that proteins are made of amino acids, and amino acids are held together by peptide bonds. So pepsin is a general protease protein that breaks down or hydrolyzes the peptide bond. Remember that hydrolysis, the word lysis means to break apart or to tear apart. The word hydro means water. So hydrolysis refers to the breaking down of things by adding water to them. Generally, when you put together two amino acids to make proteins, you remove a water molecule and you form a bond called a peptide bond between the two amino acids. If you want to do the reverse and you want to break the two amino acids apart, you add water and you include some kind of a protease enzyme that will break the peptide bond. In this case, the enzyme is called pepsin, and it will simply break down all of the peptide bonds that it can find. Okay, so the stomach is a little compartment where things are added to the alimentary canal, added to the food as it's traveling down the alimentary canal, and so protein digestion begins in the stomach. Then the gallbladder is located just underneath the liver, and the gallbladder puts in something called bile into the duodenum, which is the first part of the small intestine. And the bile helps to digest fats, lipids and fats and oils. The bile doesn't actually contain any digestive enzymes, but what it does is it's a salt. It's a salty mixture, the bile, and it has the effect of emulsifying lipids. It emulsifies fats. And if you're unsure of what the word emulsified means, it means that it just breaks it up into microscopic little beads. And then the pancreas adds other digestive enzymes to the intestine. And some of those enzymes are lipases that break down lipids. Those are fats and oils. Now, any enzyme any enzyme has an easier time breaking things down if you break it down, if you break that stuff into small parts. Just imagine that you had some wood. If you had a great big piece of wood, a log, for instance, and you tried to set it on fire, it would burn. It would be very hard to get it set on fire because it has a very small surface to volume ratio. On the other hand, if you saw that log up into sawdust and then you try to set sawdust on fire, it practically explodes because the fire has so much surface area to work on and to break down. Well, enzymes work the same way. If we break our food into smaller pieces, it's easier for the enzymes that digest our food to break them down, to hydrolyze all of them. So that's part of the reason why we chew things up in our mouth. We want to break the food into smaller pieces to give the enzymes a better chance at getting at it, to get at all of it. So when we have bile being sent into the duodenum by the gallbladder, the purpose is to break the fats and the oils into small little droplets so that the lipase enzymes from the pancreas can break them apart or hydrolyze them. The pancreas also produces a lot of proteases and polysaccharidases that break down carbohydrates and so on. Right. So the accessory organs are a bunch of organs that feed things into the alimentary canal as the food is traveling through the alimentary canal. Right. So the accessory organs are classified as the liver, which produces the bile and puts it into the gallbladder, the gallbladder, which puts the bile into the duodenum, the pancreas, which puts a whole bunch of other digestive enzymes into the small intestine, and the salivary glands, which put saliva, mix saliva in with the food, and the saliva contains amylase enzymes. Okay, and then like every other lecture, we'll finish with some discussion of pathologies of the digestive system. All right, so as I said, we have the alimentary canal. That's the mouth, the pharynx, the esophagus, going down the esophagus and into the stomach, and then from the stomach into the duodenum, the jejunum, and the ileum. The duodenum, the jejunum, and the ileum are all classified as parts of the small intestine. Then we have this little kind of sac structure here, which is the spot where the small intestine feeds into the large intestine. It's called the cecum. And then the large intestine, which is also known as the colon, it's a single continuous tube, as you can see, but for descriptive reasons, we often divide it into four different parts. The part that goes from here upwards is called the ascending colon. The part that then travels this way across the body is called the transverse colon. And then you probably guessed that going back down on the other side, on the left-hand side of the body, that's called the descending colon. And then this bottom part of the colon that's sort of shaped like the letter S is referred to as the sigmoid colon. Right, so the ascending colon, the transverse colon going from side to side, the descending colon, and the sigmoid colon, and then the anus at the bottom. That is the alimentary canal. That's the alimentary canal. The accessory organs include the salivary glands in the mouth, the liver, the gallbladder, which is located, this kind of a green light bulb thing, that's light bulb shaped structure that's located just underneath the liver, and the pancreas, which is located behind the stomach, and we'll look at that later. Okay, so let's look at the different parts of the alimentary canal one more time. So the oral cavity, that's the mouth, of course. The pharynx is the whole area associated with the mouth. The esophagus is the tube that leads from the pharynx down to the stomach. Then we have the stomach. Inside the stomach, pepsin is added to the food. You've probably remembered from the other lectures that pepsin works best at a pH of 2. So the stomach contains cells that pump hydrochloric acid into the stomach to lower the pH all the way down to 2, giving the pepsin a chance to work. So the pepsin will hydrolyze the peptide bond and thus break apart all proteins. It will begin to break apart proteins. Okay, and then the stomach, the food leaves the stomach and enters the small intestine. And we'll talk in more detail about this later, but there's a little ring of muscle tissue that closes off, kind of pinches off the bottom of the stomach. And whenever you have a ring of muscle that closes off or pinches off a tube, it's referred to as a sphincter. A sphincter. So there are two sphincters associated with the stomach. The top sphincter, this sphincter that's located at the top of the stomach, is referred to as the cardiac sphincter. Normally we associate the word cardiac with the heart, but in this case it's referring to a piece of the stomach. Right, so this cardiac sphincter, the purpose of the cardiac sphincter is to prevent all of that hydrochloric acid that's in the stomach from bubbling up into the esophagus. Right, so that prevents what's known as acid reflux. You may remember from your chemistry class that reflux is where you boil a liquid up into a tube, and as the liquid is bubbling upwards into the tube, you call that refluxing. So acid reflux is where the cardiac sphincter becomes weakened for whatever reason, and you have this hydrochloric acid that's bubbling up or oozing its way up into the esophagus and causing you a burning pain, as you would if you're burnt by any pH2 hydrochloric acid. And then the sphincter at the bottom of the stomach, the bottom area of the stomach is referred to as the pyloric region. And so this sphincter at the bottom of the stomach is called the pyloric sphincter. And it actually controls entry of the food from the stomach into the small intestine. The first part of the small intestine that makes contact with the stomach is called the duodenum. And so entry of food from the stomach into the duodenum is controlled by the pyloric sphincter. Once we're in the duodenum being the part of the small intestine that makes contact with the stomach first, there are a bunch of other holes in the duodenum. One of them leads from the gallbladder, so we can put bile into the duodenum and mix it with the food. The other one leads from the pancreas, so that we can put digestive enzymes that are produced by the pancreas into the duodenum. And the purpose of this is to mix the food with some more enzymes that will break it down and hydrolyze the food. So we added amylase enzymes from the salivary glands in the mouth, and that's where we started to break down the starch, the amylose. Then we swallowed it, went all the way down the esophagus into the stomach, where we added pepsin to start breaking down the proteins, non-specifically, in general, basically. Then we move into the duodenum. Now, we mentioned that the stomach is an acidic environment with a low pH. The pH is 2 because that's the pH where pepsin works the best. But all of the enzymes that are put into the duodenum by the pancreas they have an optimum pH of 8. So they prefer a basic environment. They prefer an alkaline environment. They prefer an area with a high pH. And so the pancreas not only dumps in all of these digestive enzymes, lipases, proteases, polysaccharidases, things that break down lipids, fats, oils, proteins, proteases, and break down carbohydrates, the polysaccharidases. These enzymes have an optimum pH of around 8. If we expect them to work, we have to suddenly raise the pH from 2 to 8. Because remember, the food that's coming out of the stomach is at a pH of 2. We have to suddenly raise it up to 8, which is a little bit of a trick. And the trick that we do in order to pull this off is the pancreas produces bicarbonate, which of course is a basic chemical. So the pancreas not only sends all of these digestive enzymes into the duodenum to mix with the food, but it also raises the pH all the way from 2 all the way up to 8 by producing a lot of bicarbonate and then putting the bicarbonate into the duodenum. Okay, and then the food travels through the small intestine, and then the part of the large intestine that connects with the small intestine is called the cecum over here. And then we have the colon, or the large intestine, and as we said, we divide that into the ascending colon, the transverse colon, the descending colon, and the sigmoid colon. The waste food that we don't digest is temporarily stored in a structure called the rectum, and then exits through a hole called the anus. Okay, the accessory organs, of course, are the salivary glands, the liver, and the gallbladder, which is part of the liver, basically. And then the pancreas, which is kind of located behind the stomach. You can kind of see where the area that I've highlighted, you can see through it. There's kind of a thing that looks kind of like a corn cob that's pointing off to the left, pointing off to anatomical left. That's the pancreas. Okay, this is just another graph showing you the alimentary canal and the accessory organs separately. So here we have the pancreas. The pancreas is located behind the stomach. This thing over here is the spleen. We've already heard about the spleen. It's not part of the digestive system. It's a filter for the blood, basically. All right, so what are the sequence of events that happen when we eat a piece of food and we chew it up and we swallow it? What are the events that happen in sequence? First thing, food enters the mouth, it gets chewed up, it gets mixed with saliva. When we chew up food and we mix it with the saliva that's produced by the salivary glands, we call it a bolus. A bolus means a bunch of stuff altogether. Means a big chunk altogether. Right, so the teeth are important. The teeth are a very important part of the digestive system. They're often classified as part of the digestive system. And the purpose of having teeth is to grind up food. What was the original purpose of having a tongue? All other animals, all other mammals except humans use the tongue to position the food on the teeth so you can chew it up. So you have this little kind of a finger or this little kind of a hand structure inside your mouth that can take the food and put it on top of the teeth, and then you clench your teeth, you lower the mandible and you crunch it up, and then your tongue repositions it again, and then you crunch it up again. That was the original purpose for the tongue. We are the only mammals that used it for another purpose, which is to speak, but it was originally by all the other animals, it's used simply to position food on the teeth. And why do we want to grind the food up on the teeth? Because we want to make it into smaller pieces, because it's easier for the enzymes that are going to hydrolyze the food to get at it if we break it into smaller pieces. If you have large pieces of stuff, remember that enzymes work from the surface inward. And so if you have large bits of food, for instance, the ratio of the surface area to the volume is quite small. If you break the food up into small pieces, the ratio of the surface area to the volume is large, and therefore, in other words, there's a lot more surface for the enzymes to get at, and that's the purpose. Right. Now, the salivary amylases are added to the food in the mouth, and they are sometimes referred to as talin. But the salivary amylases is a perfect way to say it. All right, now when we swallow the food, it goes down the esophagus to the stomach using a type of pumping action called peristalsis. Peristalsis is where peristalsis is a common form of movement of things through the alimentary canal. And remember that the alimentary canal, the esophagus, the stomach, the intestines, it's a long continuous tube that is surrounded on the outside by smooth muscle. And the smooth muscle, of course, is controlled by the autonomic nervous system. And what it does during peristalsis is the smooth muscles will contract or squeeze behind the food, pushing the food forward down the canal. And then as soon as the food moves forward, the bolus moves forward a little bit, then the smooth muscle that's just behind the bolus contracts and squeezes it forward a little bit more, and it travels down the alimentary canal that way. So here we have an illustration of peristalsis. There's our bolus, right? And we squeeze, the autonomic nervous system squeezes. Let's say this is in the esophagus, for instance. The esophagus, the smooth muscles will squeeze behind the bolus, pushing the bolus forward. Right? And then it moves forward, you squeeze the muscles behind it again, you cause it to move even farther forward, you squeeze again, farther forward. That is referred to as peristalsis. All right, so the stomach, once the food gets in the stomach by being sent down the esophagus, we get into the stomach. The stomach contains hydrochloric acid, so the pH is 2, and it contains an enzyme called pepsin, whose job it is to hydrolyze, break down the peptide bonds that hold proteins together. If you take the mixture of hydrochloric acid and pepsin together, it is referred to as gastric juice. Right? And when you mix food with gastric juice, we rename it chyme. Chyme. Remember that in biology, a lot of the biology words came from Latin root words, and the Romans didn't have a soft C. They didn't use the sound ch. Everything was ka. Right? So we're used to saying the name Julius Caesar. The Romans didn't actually have a J sound, and they didn't have a soft S, Caesar. So his name in Latin was actually Iulius Caesar. But then when the Europeans got a hold of the Latin language, we kind of messed it around a bit, and so changed it a bit. So we added the ch and the s sound associated with C's. But in biology, we don't do that. We usually try to remain loyal, as much as possible, remain loyal to the original Latin. So chyme is the word for what you get if you mix gastric juice with the food, which came down as a bolus, a single large piece down the esophagus. Okay, then, so we mix the pepsin and the hydrochloric acid and the food together to form chyme in the stomach, and then digestion of a lot of the proteins takes place in the stomach, but it's often the food is not in the stomach long enough to digest all of the protein. It then moves into the duodenum through the pyloric sphincter, which I mentioned before. So we enter the small intestine, and once in the small intestine, the pancreas adds a bunch of other digestive enzymes as well as bicarbonate. The purpose of that is to raise the pH in the intestines to 8, and so therefore we say the intestines are an alkaline environment. Then the liver has produced a bunch of salts called bile salts. Bile salt is one of the major components of bile salts is bilirubin that's made from the breakdown of blood. But nevertheless, it's a salty mixture. It's kind of green and bile. You've heard the word bilious. The word bilious means a kind of a vomit green color, kind of a pea soup green color. You'll see if people vomit, the vomit often looks a little bit greenish yellow, yellowish green, and that is the color bilious. And so bile salts, bile is meant to be that color. Bile gets its name because it's that color, or maybe the color gets its name from bile. I have no idea, but that's the idea. Bile is kind of greenish. The liver produces bile salts, but it does not store the bile salts. The liver produces the bile salts and then it puts it into the gallbladder. The gallbladder is green in color. It's kind of shaped like an upside down light bulb, and it's located just underneath on the posterior, just underneath on the inferior side of the liver. And the purpose of the bile salts is to emulsify fats. In other words, break the oils and the fats that we've eaten up into small little droplets so that the lipase enzymes can digest it more easily. Okay, so here's the structure again. There's the liver, right? There's the gallbladder behind the liver. And the gallbladder is attached to the duodenum, so is the pancreas. The pancreas is visible just behind the stomach here. So the food continues to break down as it passes through the small intestine, and nutrients are absorbed into a network of veins that are referred to as the hepatic portal system. Now, the hepatic portal system refers to a whole bunch of veins that surround the outsides of the small intestine. And so in the small intestine, proteins are broken down into amino acids, lipids are broken down, and lipids and fats and oils are broken down into fatty acids. Polysaccharides are broken down into monosaccharides, right? So then those amino acids, those monosaccharides, and those fatty acids are absorbed through the wall of the small intestine, and then they are absorbed by a bunch of veins that are on the outside of the small intestine these veins are collectively referred to as the hepatic portal system. The hepatic portal system veins all converge into a large vein called the hepatic portal vein, which is a very large vein, which leads from the small intestine into the liver. Right. And then all of that food that we absorbed in our small intestine it gets into the blood through these hepatic portal veins, but it is not allowed to go into the general circulatory blood until it's been filtered and detoxified. What do I mean by that? I mean when we break down proteins, we often end up accidentally breaking off the amino the amino group from the amino acids, and that leads to the production of ammonia, and ammonia is a very toxic substance. So we get rid of the ammonia. The ammonia is usually broken down and it's usually converted into urea, a less toxic compound called urea in the liver. And we're going to talk about the conversion of ammonia to urea later when we discuss the renal system. But generally, there's a lot of ammonia that's generated when we eat food and digest it and break it down, and there's also a lot of peroxide that's generated when we chew up the cell. If you eat animal flesh, for instance, every cell that the animal flesh is made of contains mitochondria. Mitochondria tend to produce a lot of hydrogen peroxide as a waste product, and that's toxic as well. Alcohols get produced here and there, and so the alcohols, the ammonia, the peroxide, all of that stuff has to be detoxified. Those are all toxic waste products that result from the digestion of food. And they have to be detoxified before you can allow that blood to go into the general circulation. So we have to clean it up first. So the blood does not, all of these nutrients that are absorbed by the intestines and they're absorbed into the blood, they are not connected to the general blood supply yet. They have to pass through a filter before they can go into the regular blood. So the liver is the filter. So we have all of these veins that take blood from around the small intestines. They converge into a single vein called the hepatic portal vein. As you know, the word portal means an entrance to something. So all of these veins converge into the hepatic portal vein, which goes into the liver. The liver then detoxifies all of these products, and then it allows the nutrients, once they've been detoxified, the nutrients will then enter the regular blood supply. So in fact, what happens is we have the hepatic portal vein taking the food from the intestine into the liver. That is called the hepatic portal vein. And then once it's detoxified, it takes the blood from the liver to the inferior vena cava. And that vein is called the hepatic vein. So I'll warn you, don't confuse the hepatic portal vein with the hepatic vein, because they're two very different veins. They're traveling in the same general direction. They're traveling from the intestine to the inferior vena cava, but they have to go through a filter first, right? So the hepatic portal vein leads from the intestine to the liver. The hepatic vein leads from the liver to the inferior vena cava. So the blood that is in the hepatic portal vein is full of toxins. The blood that's between the inferior vena cava and the liver is cleaned up. Okay, now what about the large intestine? The large intestine has a number of functions, but probably the most important one is that it is there to recover water. Remember that in order to digest food, we have to add water to it. That's why we call it hydrolysis. So if you look at food when it's in the stomach, it's quite moist. It's kind of like a soup. It's very runny. If you look at food as it's going through the small intestine, it looks like diarrhea, basically. It looks like a brown soup, right? And that's because the food is mixed with a lot of water. Okay, now as you know, if you have diarrhea, the food is coming out when it's still watery, because usually diarrhea happens because you've eaten something you're not supposed to, and your body says, get it out of here. As soon as it gets into your intestine, the intestine says, oh, that's awful, get it out of here fast. I don't care about losing all that water, just get it out. But under normal circumstances, if you eat regular food, it is mixed with a lot of water as it's going through the small intestine. And then we don't want to lose all that water because we'd have to drink an awful lot to replace it. So the large intestine, one of its main functions is to recover the water. It soaks a lot of the water back out of the food, so the food becomes more and more like a solid. And as you know, excrement comes out, it's a soft solid. You know what it's like when it comes out, it's a soft solid. Whereas if you looked at it while it was in the small intestine, it would be like a brown soup, And so one of the main purposes of the colon is to recover the water so that we're not constantly having to drink water to replace it. Now, along that same line of thought, if someone has a disease that causes diarrhea, one of the ways that people often die from diseases is by getting really bad diarrhea. Cholera, for instance, you get really bad diarrhea. Typhoid, you get bad diarrhea, things like that. And you often die from dehydration. The humans are not able to not able to survive if they lose a lot of their water budget. When we talk about the renal system, we'll learn that the water budget is about 2.5 liters of water a day. Generally, we lose about 100. So we drink about 1.5 liters of water a day. We get about 100 or 250 mils of water every day from eating our moist foods, vegetables and fruits and whatnot. And then we also produce some water when we breathe, because the respiratory system generates water, believe it or not. And so we have to have a water budget of around 2.5 liters a day. And then when the water comes out the other side, we generally urinate 1.5 liters a day. We excrete in our feces. We lose about 100 mils of water. If you have chronic diarrhea, you might be losing 500, 600, a liter, or even a liter a day of water. You can see that if you're losing 10 times more water in your excrement as you're supposed to be, you either have to compensate by drinking a lot of water, a lot of fresh water to replace it, and if not, you'll dehydrate and die. And often a lot of these diseases like cholera and typhoid fever or amoebic dysentery, which is caused by Entamoeba histolytica, causes diarrhea. People die from these diseases because they get a very bad case of diarrhea and they dehydrate. And so people end up having heart attacks and dying or because they have a lack of water. All right, so here we have the intestine. This is a shot of the small intestine from the inside. And we've discussed this theme before in this course, that if you have a membrane that's important, something really important happens at the membrane, like the cerebral cortex, for instance, or the lungs. The cerebral cortex, we have gray matter neurons, or the lungs, we have gas exchange happening. And in the intestine, we're absorbing food. Right? So the inner membrane, the inner surface of the intestine is made to absorb these nutrients, and the larger it is, the more we can absorb. Therefore, the larger it is, the more efficient it is, the larger the surface area. And we've discussed this theme again and again, the fact that if you need to have a large surface area in a small space, you get away with doing this by folding it. That's why the cerebral cortex is so heavily folded, because if you stretched it out so it was flat and not folded, it would be quite large. But we have to fit it into this relatively small space, the cranial vault. The same thing is true of the intestines. So we have this tube, and if you look at a worm, for instance, a worm is a very primitive animal, and it's basically just a mouth and an anus with a long intestine in between, and the intestine is as long as the worm is. Right? But the human intestine is folded and folded and folded. The tube is folded folded many times so that it can fit into the abdominal cavity. But it's still quite large. It just doesn't look that large because it's been folded. In fact, if you unfolded it and stretched it out, it would probably be about 6 times taller than you are. So the intestine is actually quite large. And then furthermore, if you look at the inner surface, it's folded, and the more folds you have, the more nutrients you can absorb, so the more efficient a feeding system it is, the more heavily folded it is. Right, so the inner surface of the small intestine, it's pink in color, right? So in some of our courses here at Columbia, we dissect rats and rabbits and things like that, and you look at the intestine and it's gray in color. That's only because the animal is dead, and so there isn't blood circulating through it. But if you look at a live intestine, it's pink on the inside, and the surface is folded into these folds that are called villi. And then if you look at the individual cells that make up each one of these villi, it's even the cells themselves have little folds as well that are referred to as microvilli. So we'll talk about that later. And then the whole purpose of that, of folding the tube into many times into a little space, and then folding the surface of the inside of the tube, and then folding the surface of the surface, and then folding the cells that form the surface. Folding. The purpose is to take a very large membrane and squeeze it into a small space, but it's very efficient. It's a very efficient way to absorb nutrients. And then here we have a network of veins. The veins carry the blood to the liver. That network of veins is collectively called the hepatic system, or the hepatic portal system rather, and they all converge into one large vein called the hepatic portal vein. So here you can see the hepatic portal vein leading into the liver, and then here we have the hepatic vein leading out of the liver and heading towards the inferior vena cava. Okay, so let's look in detail at some of the structures that are in the oral cavity. The oral cavity is also known as the buccal cavity. Remember, on the first day of class, we discussed, we did that exercise, and we learned that the area inside the mouth is referred to as the buccal cavity, and the inside of your cheeks are lined with a type of cell called a buccal cell. these buccal cells are loosely packed onto the inside of your cheeks so that they can be easily torn away by rough food that you might have eaten. Right? So to avoid tearing the tissue of the mouth, the inner surface of the mouth is covered with these buccal cells that are easily torn away, so that if the food is going to scratch the inside of your mouth, the worst thing that will happen is it will tear away some of the buccal cells, which are meant to be torn away and replaced by the time you have lunch, right? Basically. Okay, so, all right, so the buccal cavity. First of all, we have a pair of lips. The Latin word for lips is labia, right? So we have a superior and an inferior labia. Then we have the teeth, right? The teeth are for grinding up food. The technical term for grinding up food is mastication. Mastication, right? The study of teeth is referred to as odontology. Right, so there are a lot of scientists who look at the fact that, you know, carnivores have long fangs called canine teeth, and herbivores do not have the canine teeth. And so the study of the shape and function of teeth is called odontology. It's an important field of research, but that's what it's called. And then we have the buccal cells that line the inside of the buccal cavity that are easily torn away when we swallow our food. By the way, I've mentioned this before in class, but when they get a DNA sample from you to do a DNA fingerprint, either they're trying to determine your ancestry, or they're trying to determine whether you're a murder suspect in some horrible crime or something like that, they ask for a DNA sample. They take it from the buccal cells. And what they do is they either get you to spit in a tube, and your saliva, your spit, will contain a lot of buccal cells. And the buccal cells have nuclei, so you can get the DNA out of the nuclei. And or they will take a swab, you know, like a Q-tip, and they'll rub the inside of your cheek and then put that into a little tube and then do a test called a PCR test, a polymerase chain reaction test, to do DNA fingerprinting. So that's usually where they get a good source of DNA to do DNA fingerprinting are the buccal cells. Okay, so then as we said, the tongue, the purpose is to position food. Then we have the lingual frenulum. The lingual frenulum is that little flap of tissue that's underneath the tongue that stops us from swallowing our own tongue. So the reason we might accidentally swallow our own tongue, if not for that little flap of tissue called the lingual frenulum. And then the front part of your mouth is referred to as the hard palate because it's right underneath the maxilla. And then the back part of your mouth is called the soft palate because it's basically just starting to turn into this muscular tube and doesn't have any bones above it. All right, so here's a diagram of the oral cavity, right? So there's the lingual frenulum right there. Right? There's the tongue, of course. This little punching bag thing, it always looks like a punching bag that a boxer would use. That's called the uvula. Right? That's the thing that makes the noise when you snore. Right? So these teeth at the back are referred to as the molars. You learned about that in the lab. And then the little fangs, or the kind of pathetic excuse that we have for fangs in humans, are called cuspids. In animals, they're called canines. Those are in a dog or a tiger. They're quite large. Right? And then we have some smaller molars called premolars, and then we have some teeth. We have the cuspid teeth, and then in the front here we have the incisors that are meant for biting things off, and then the molars are meant for grinding them up. Okay, what are the canine teeth for? Canine teeth are usually absent in herbivores, animals that eat vegetation, but the canine teeth are usually quite enlarged in animals like tigers and things because their main use is for killing. In fact, that if you see a tiger killing an animal, the first thing it does is it takes its big canine fangs and tears into its neck, tears into the prey animal's neck, and that usually tears the carotid artery in the jugular vein, which kills it fairly quickly. So the purpose of large canine teeth on carnivorous animals is to kill the prey animal quickly, because otherwise your supper keeps getting up and running away. So that's not very good. So you want to kill it as quickly as you can. Okay, there's our friend, the lingual frenulum. All right, just looking at a sagittal section through the skull. Right, so the palatine bone is located here, which forms part of the hard palate, and then most of the hard palate is formed by the maxilla. Okay, so as I said, the uvula is that little thing that looks like a boxer's punching bag. That's what's making all the noise when you snore. The palatine tonsils, commonly known as just the tonsils, are part of the immune system. They are basically a kind of a lymph node, which we talked about when we discussed the lymphatic system. Okay, then we have a bunch of salivary glands. Right, so we have three sets of salivary glands. So 3 times 2, one on each side. So we have a left and right parotid gland. These are located nearest the ears. They produce saliva. Then we have a left and right sublingual gland. Where do you think they are? Under the tongue, of course. The tongue referring to lingual region. And then we have a pair of submandibular glands, which of course are under the mandible. So they're located under the jaw. And combined, these 6 salivary glands, these 3 sets of salivary glands, produce an amazing 1.5 liters of saliva a day. Now that's a lot. It doesn't seem like a lot because we're always swallowing it, but that is a lot, right? And it doesn't really matter because you're reabsorbing it in the intestine anyway. You're reabsorbing it in the colon, so it's not like you lose it. But the salivary glands do have to actually, they need to take a bunch of that water, they take it out of the blood serum, and then the salivary glands produce the amylase enzymes and a few other things, and they put it into let it go in the mouth so that it can be mixed up with your food. So when you chew up your food, you mix it with saliva, and then you rename it as a bolus. All right, here are the salivary glands. The parotid glands are closest to the ear. The submandibular glands are located underneath the mandible, underneath the jawbone, and the sublingual glands are located under the tongue. If you reach around, if you feel around inside your mouth with your tongue, you can usually feel the hole. There's a little flap that empties from the parotid gland into your mouth, and you can actually see where the saliva from the parotid gland enters the mouth. You can actually feel it with your tongue. So this is the parotid duct. As I said, you can feel that with your tongue. Right? It comes into your mouth sort of just above the molars. All right, now the area behind, sort of back in the throat. So if you look at the hollow area behind the buccal cavity is referred to as the pharynx. And we sometimes divide it into the nasopharynx versus the laryngopharynx. Right? Laryngeo refers to the larynx, which is commonly called an Adam's apple. It's a lump of cartilage that you can see in your neck on the front of your throat. And then there's a hollow area where, remember that food goes down the esophagus, and when we breathe, the air goes down the trachea. There's a common compartment where both the trachea and the esophagus empty, or the intake for both the trachea and the esophagus are in the glottis, right? So there is a flap of tissue, a flap of connective tissue called the epiglottis. Remember, the word epi means above, and so the glottis is this kind of a chamber where the entry to the trachea and the entry to the esophagus are combined. And we have a flap of tissue called the epiglottis, which is critical to a function called the swallow reflex. Now you notice that when you swallow, you can't breathe. And the reason for that is because the epiglottis has closed like a lid to cover the top of the trachea, leaving the esophagus open, so the food will go from your pharynx down into the esophagus, as opposed to any of the food or any of the drink going into the trachea and causing you to have a coughing fit. So that is referred to as the swallow reflex, and that's a function of the epiglottis. All right, so the glottis contains the entry to both the esophagus and the trachea. Right. And at the very top of the esophagus, there's a sphincter, a ring of muscle that closes off the top of the esophagus after you've swallowed, and that's called the esophageal sphincter. Right. And then the food goes down the esophagus via peristalsis, peristaltic pumping. Okay, so we swallow the food, it gets into the nasopharynx, and then here we have the glottis, See the glottis here? So the glottis contains the entry to both the esophagus and the trachea. The trachea leads to the lungs. We don't want the food to go that way. We want it to go down the esophagus this way. Now, if you ever, some of you have dissected rats in Biology 120, for instance, you know that if you cut open the throat of the rat, you will see both the trachea and the esophagus. And how do you tell them apart? Well, you tell them apart because the trachea is being held open by rings of cartilage, and so it's open, whereas the esophagus is usually collapsed because it's, there's nothing inside it. There's no reason for it to be open. All right, so here we have the glottis, and then here's a flap of connective tissue called the epiglottis that closes off the top of the trachea to prevent our food from going down to our lungs. All right, looking at the larynx is this bump that's mainly made of cartilage, and the cartilage is given different names depending on what position it is. So here's an anterior. On the left, we have an anterior view of the larynx. Right, so we're looking at it from the front, and this bump that we call the Adam's apple is located right here. And it is made of a type of tissue called thyroid cartilage. And the reason it's called thyroid cartilage is because the thyroid gland is sitting right on top of it, basically, or sort of underneath it. And we'll talk about the thyroid gland when we talk about the endocrine system, but that's why it's called thyroid cartilage. And then underneath we have cricoid cartilage, and then we have a bunch of rings of cartilage that are holding open the trachea so that it won't collapse under negative air pressure. Okay, so the cricoid cartilage, by the way, the cricoid cartilage is the dividing line. We talked a little bit in this course about anatomical landmarks and how do you divide what is the landmark that separates the thorax from the abdomen? It's the diaphragm. Okay, what is it that divides, what landmark do we use to divide the upper respiratory tract from the lower respiratory tract? And the answer is the cricoid cartilage. So this band of cartilage here is the official dividing line between the upper respiratory tract and the lower respiratory tract. All right, so if we have a posterior view, of the larynx, notice that here we have vocal cords, and here we have the epiglottis, whose job it is to kind of stand open when we're breathing and then slam shut when we're swallowing food. That's called the swallow reflex. Okay, so if we're about to swallow a bolus of food, the epiglottis is open. All right, so we swallow the food, the epiglottis slams shut so that the food can go by without entering the trachea. And of course, the esophageal sphincter has to open in order to let it go by, and then it usually closes again once the food has gone past. And then the food travels down into the stomach using peristalsis. And I mentioned the fact that the esophagus is bounded by two different sphincters. At the top of the esophagus, we have the esophageal sphincter. So it opens to let the bolus go from the mouth to the esophagus, and then it closes again once it's gone past. And then we have the cardiac sphincter, which is located at the top of the stomach. The purpose of the cardiac sphincter is mainly to prevent hydrochloric acid from refluxing up into the esophagus. Okay, so don't confuse. There are actually three sphincters kind of in close proximity. The esophageal sphincter is located at the top of the esophagus. The cardiac sphincter is located at the top of the heart. And what was the other one again? Located at the bottom of the stomach. That's called the pyloric sphincter. So don't confuse where those three are. I often on final exams, I'll ask students where is the esophageal sphincter located, or what do you call the sphincter that closes off the top of the stomach? And they'll answer esophageal sphincter, but that's wrong. That closes off the top of the esophagus, not the top of the stomach. Okay, so we talked about peristalsis. It's a common way of moving things through a tube. If you ever end up working in a hospital or any other kind of a thing where you have access to certain types of pumps where you're trying to pump liquid through a hose, this type of a pump is called a peristaltic pump. And look at how it works. It works because you have there's a little electric motor and you have some rollers here, and those rollers are squeezing this piece of rubber hose in the same direction, that way. Right? And that's how a peristaltic pump works. So if you have an IV bag for a patient or something like that, this is a peristaltic pump. So there's a rubber hose that comes from the IV bag. You have to hook it into the peristaltic pump, and then you have a venipuncture site usually on the dorsal side of the hand, and then the IV bag will empty into the patient that way using a peristaltic pump. All right, so looking at the inside of the stomach, we said that the top of the stomach is closed off by the cardiac sphincter. The cardiac sphincter obviously has to open to let the food bolus in, and then it closes again to prevent stomach acid from bubbling up into the esophagus and burning it. You might ask, how does the stomach avoid getting burnt by its own hydrochloric acid, and how does it prevent getting digested by its own pepsin? Good question. It does this because it is a mucosa. The layer lining of the inside of the stomach is columnar epithelium. It's a mixed heterogeneous columnar epithelium that contains a whole bunch of goblet cells that produce mucus. And so the lining of the stomach is a mucous membrane or a mucosa. So it's made of columnar epithelium with a few goblet cells, and it produces a thick layer of mucus. You know what mucus is, of course. That's what the stuff that runs out of your nose when you have a cold. It produces a very thick layer of mucus that prevents the stomach lining from being burnt by its own hydrochloric acid. Okay, now the different parts of the stomach have names. The part near the cardiac sphincter is named the cardia. And then the upper bulge of the stomach is called the fundus. Then we have the body, and then the bottom part of the stomach is the pylorus. The inside of the stomach is folded many times to allow it to stretch. Right, so you know that when your stomach is empty, it occupies a small space, and then when you've eaten a huge meal, you have to undo your belt because the stomach is temporarily filled with a lot of food. That food eventually goes into the intestine, but at the beginning it's being held in the stomach. And so the folds inside the stomach are given a special name. They're called rugae. Okay, now if you look closely at the surface of the stomach contains some little pits that are referred to as gastric glands. The word gastric refers to the stomach specifically, gastric glands. And inside a gastric gland, you will find regular columnar epithelium. You might find a few goblet cells, but in the gastric glands, you will find a type of cell that's referred to as a chief cell. The chief cells produce the pepsin enzyme. And in fact, when the chief cells produce pepsin, they produce it initially as an inactive precursor called pepsinogen. Okay, so pepsinogen is a polymer where you have multiple pepsin enzymes that are held together in tandem, one after another. And they are inactive. When they're all hooked together like that, they're inactive. If you want to activate them, you have to chop them up into individual pepsin enzymes. What is the enzyme that chops up the pepsinogen into pepsin? It turns out that it's pepsin itself. Right? So if you have some pepsin left over in your stomach from breakfast, your stomach the chief cells in your gastric glands will start to produce pepsinogen by lunch, and then that pepsin that was left over from breakfast will chop up the pepsinogen and activate it. Okay, and that's not enough. As we said before, the pH needs to be low at 2, right? So we have in the gastric glands, we also have a type of cell called a parietal cell that produces hydrochloric acid to lower the pH. Right, so we mentioned the fact that if you have pepsin mixed together with hydrochloric acid, that's referred to as gastric juice. As soon as you mix the food with it, we call it chyme. So you mix a bolus of food that already has saliva. You have saliva and food, that's a bolus going down the esophagus. The bolus enters the stomach, it mixes with gastric juice, and we rename it as chyme. Okay, looking at the stomach here, So here's the esophagus up here at the top, and then we have the cardiac sphincter, which closes off the top of the stomach. The area of the stomach that's near the cardiac sphincter is referred to as the cardia. This kind of bulgy part of the stomach is called the fundus. The main part of the stomach is called the body, and then the bottom part of the stomach is called the pylorus. And then at the base of the pylorus we have the pyloric sphincter, and then we have the small intestine leading on from there. That is the duodenum. And you can see that the stomach is heavily folded into rugae, and then outside the rugae you have a bunch of smooth muscle that's being controlled by the autonomic nervous system. There's several layers of smooth muscle that kind of run in different directions. The stomach has a lot of power to kind of contract and squeeze the food. That's kind of to mix it. So when you eat food, your stomach makes that growling, gurgling sound. That's the stomach, these smooth muscles that are pointing in different directions, kind of squeezing this way and then squeezing that way in order to mix the food with the gastric juice. All right, I'm going to show you an image of a human stomach, so if that disturbs you, look away. But you'll see the rugae in real life. All right, so once again, here we have the cardiac sphincter, pyloric sphincter, right? The cardia, the fundus, and the body, right? And then we have all these rugae that allow the stomach to expand when it's filled with food. So that's the purpose of having these folds, is to allow the stomach to expand to hold a lot of food. All right, as we said, you can then have, if you look closely at the surface of the stomach, you will find these little pits in the stomach that are referred to as gastric glands. If you take a close look at the gastric glands, you'll see that they go way down. These little things here are all individual cells. And if you look at the individual cells, you have chief cells that produce pepsinogen. Pepsin is the active form of pepsinogen. Pepsinogen is converted into pepsin by pepsin itself. Right? And then we have parietal cells that produce the hydrochloric acid that allows the pepsin to work. The optimum pH for pepsin is pH 2. So as soon as you swallow food and it goes into the intestine, largely the pepsin that came with it from the stomach is not working very efficiently because it's at a very high pH by the time it leaves and goes into the duodenum. Just another diagram of a gastric gland showing parietal cells and chief cells. Right, so here we have the gastric glands, and then we have the layers of smooth muscle that are running in perpendicular to each other so that the stomach can squeeze in one direction and the other. The different layers of smooth muscle are referred to as the oblique layer, the circular layer, and the longitudinal layer. Okay, so we have the gastric glands. We have the pyloric sphincter located at the bottom of the stomach, which guards and controls entry into the duodenum. So the stomach allows the food to enter the duodenum a little bit at a time so the intestine doesn't get overwhelmed. And that's controlled by the pyloric sphincter. And so there's the pyloric sphincter. There's the cardiac sphincter. And this is all controlled by the parasympathetic nervous system, by the vagus nerve. Okay, so the whole process of digesting food is controlled by a number of different enzymes. Remember that enzymes are part of the endocrine system. The stomach is technically not part of the endocrine system, but it does produce some of the hormones that it needs to do its job. Okay, so 3 hormones for this introductory course. We'll memorize 3 of the hormones. Right, so to begin with, we have the hormone called gastrin. So gastrin is produced by a type of cell called G cells in response to proteins being in the stomach. So as soon as you swallow proteins, the G cells get busy and they produce gastrin. And that hormone increases the secretion from the gastric glands and stimulates peristalsis in the intestines. So it tells the intestines, okay, you've got old food in there from breakfast, move it out because we're going to get ready and put some new food in that has more nutrients in it. You're done with the food from breakfast, just move it out of there. And it also increases the blood flow to the intestines because they're going to have to do some work. So those are functions of gastrin, the gastrin hormone. All right, if you haven't eaten for a while, there are cells in the stomach that produce an enzyme called ghrelin, which makes you feel hungry. It's produced in response to fasting or abstaining from eating for a while. So usually when you wake up in the morning, you haven't eaten for 10 hours or 12 hours, there's a lot of ghrelin being produced. Okay, and then if gastrin turns the gastric glands on, somatostatin turns them back off. So they turn the gastric gland secretion back off again and reduces blood flow to the intestine. This is presumably after we've eaten all our food, it's been through the stomach is now empty again, we don't need all the gastric glands to be producing all of this pepsin and hydrochloric acid, turn it back off again. You probably also know that coffee contains caffeine. Caffeine stimulates the gastric glands as well, so that if you drink coffee right after a meal, that actually does help digestion. So that's coincidentally why a lot of people drink coffee after eating a meal. However, if you go on drinking coffee like I do until 1 or 2 in the morning, I love my coffee. I drink it all day long, and as a result of that, I have a lot of acid reflux. I often get heartburn, acid reflux, because I drink coffee late into the night. And then when I lay down to go to sleep, the hydrochloric acid bubbles up through the cardiac sphincter and then causes heartburn. Okay, so we said that the small intestine it's really a continuous tube, but for descriptive reasons, we divide it up into the duodenum, which is the first part, the jejunum, the second part, and the ileum, the third part. And then there's a valve that leads to the cecum that connects the ileum to the cecum, and that's referred to as the ileocecal valve. Also sometimes referred to as the ileocecal sphincter. Then we have this little kind of an enlarged sac structure that's called the cecum. The appendix is hanging off the bottom of the cecum. The appendix is a vestigial immune organ. Vestigial means it's something that we used to use and we don't use it anymore, so that organ has kind of disappeared. So the word vestigial, you'll hear it a lot. For instance, if you look at a snake, if you take the skin off of a snake, you'll notice that it has shoulder blades. Snake has little vestigial shoulder blades, even though the snake doesn't have arms. So we're assuming that at some point during its evolution, the snake did have arms, and it doesn't have them anymore. So if you don't have arms, what do you need scapulas for? So a snake has vestigial scapulas. But anyway, the word vestigial. The appendix is a vestigial immune system organ. So there are immune system cells in the appendix. These days we don't use it for that anymore. It usually just ends up being a place where the excrement gets accidentally diverted, or the appendix gets broken, or it gets inflamed and causes a lot of problems. And then we have what's known as an appendicitis if the appendix gets inflamed and swollen up and causing a fuss. And then you have to have an appendectomy. You get the appendix taken out. Okay, so the cecum is the doorway to the large intestine, guarded by the ileocecal valve, and then the food goes into the large intestine, the colon. All right, so here the purple is the duodenum. That's the first part of the small intestine that makes contact with the stomach. The bluish part is the jejunum, and then the pinkish part is the ileum, and then through the ileocecal valve we're connected to the cecum right there. All right, let's look at the small intestine from the inside out. Right, so there are 4 layers of it. Okay, so the layer that's facing the food, the innermost layer that's lining the lumen, is a mucosal membrane. So it's columnar epithelium that has some goblet cells to produce mucus. So the mucosa that lines the innermost, the lines of the lumen of the small intestine, is folded into villi. And then the individual cells that make up the villi are folded into microvilli to increase the surface area. Each villus has its own capillary underneath it to supply it blood and to absorb nutrients. And there's also, each villi has a lymphatic duct or a lymphatic vessel called a lacteal. Lacteal, right? So every villi, the individual folds of the intestine are called villi. Each villi has a capillary and a lacteal. Then just underneath the mucosa, you have a layer that's not surprisingly called the submucosa. It's made of connective tissue and it has some blood vessels in it. And then beyond that, we have the muscularis externa, or the smooth muscles that are wrapped around the outside of the small intestine that are involved in the peristalsis. And then we have a layer of tissue called the serosa, which is basically the inner layer of a serous membrane. So remember that you have a serous membrane that's surrounding the intestines, right? We talked about that earlier. And the serous membrane that's surrounding the intestines, the peritoneum, has like any serous membrane has two layers, and the inner layer is referred to as the visceral layer, and the outer layer is referred to as the parietal layer. And so in this case, because the serous membrane is surrounding the intestines, and that serous membrane is given a special name, we call it the peritoneum. The serosa is actually the visceral peritoneum. Just another diagram. So we have the mucosa on the inside, right? The epithelial, made of columnar epithelial cells. And then we have muscularis, layers of muscle. And then we have the serosa. Okay, so here we have a close-up of the villi, right? So the internal structure of the small intestine, it was folded to begin with. And then if you look at the individual folds, you see that there are folds, little finger-like folds within folds that are called villi. So these things are all referred to as villi. Each of these little squares inside the villi is a cell, and if you look at the individual cells, you see that they have microvilli. So even the plasma membrane of those cells is folded into little finger-like structures. And the purpose of that whole thing is to increase the surface area so we can absorb more nutrients. The nutrients are absorbed into the capillary that's located inside each villa, and at the center of that capillary there's also a lymphatic duct called the lymphatic vessel called a lacteal. All right, just some other diagrams. If you look at a histology section, you look at a cross section, this area here where you see the microvilli on the surface of the villi, that is referred to sometimes as a brush border. So here you see the villi, so that the area where you see these villi is sometimes referred to as a brush border. All right, so the duodenum, a very special part of the small intestine, the first part to make contact with the stomach. So the food, the chyme, comes out of the stomach through the pyloric sphincter. The entry is regulated. Entry into the small intestine is regulated by the pyloric sphincter. And in the duodenum, the pancreas puts in bicarbonate, and it puts in digestive enzymes. The digestive enzymes include lipases that break down lipids, fats and oils, proteases that break down more of the proteins, nucleases that break down nucleic acids, polysaccharidases that break down other polysaccharides. And the pancreas delivers all these enzymes to the duodenum through a tube called the pancreatic duct. The liver produces a compound called bile, a salty green substance that's referred to as bile or bile salts, but it doesn't store it. It puts it into the gallbladder, this light bulb shaped thing, and then from the gallbladder it travels to the duodenum through a tube called the common bile duct. So there's the pancreas behind the stomach, and there's the gallbladder underneath the liver. This is a schematic diagram of the digestive system. So instead of showing you the anatomical pictures, I'm just showing you kind of a circuit diagram. So here we have the alimentary canal consisting of the mouth, the esophagus, the stomach, the small intestine, the large intestine, the rectum, and the anus. It's a single continuous tube. The accessory organs are sending things into that tube. So here we have the salivary glands putting in saliva, which contains amylases to begin the digestion of starch. The stomach produces its own pepsin, which begins the digestion of proteins inside the stomach. And then we have the liver, which produces bile salts, which are stored in the gallbladder and then put into the duodenum. And we have the pancreas, which is producing bicarbonate and various digestive enzymes that are being put into the duodenum as well. So here is a diagram showing the gallbladder if we remove the liver, so you can't see it. So there's the gallbladder sending bile salts down into the duodenum. This thing here is the duodenum. And then here we have the pancreas sending bicarbonate and digestive enzymes into the duodenum. And then the food is entering the duodenum from the stomach through the pyloric sphincter. So we're mixing in the duodenum, we're mixing the food together with the digestive enzymes, the bicarbonate, and the bile salts. Okay, so once we've added all that stuff to the food, they continue their journey through the small intestine. The food continues to be broken down and hydrolyzed as it travels through the jejunum and the ileum. The nutrients are absorbed by the veins of the hepatic portal system, and then they're sent to the liver for detoxification. Then the food enters the cecum through the ileocecal valve. And now we're in the large intestine. And the main purpose of the large intestine is to recover the water. Right, so hydrolysis of food requires that we add large amounts of water to the food, but that's okay. We're not going to lose it. We're going to take it back. We'll reabsorb it in the colon. Another thing that happens in the colon is that the colon is also important for absorbing vitamins and minerals. And also we have a whole bunch of vitamins and minerals, for instance, vitamin K, calcium, and so on. And we also have a whole lot of symbiotic bacteria that live in our colon. One of which is called E. coli, Escherichia coli, and Escherichia coli is named after the fact that it is found, among other places, in the colon. It was first discovered in the colon, but it's it's everywhere basically. And Escherichia coli produces more vitamin K than it actually needs. And so the vitamin K that is being produced by the E. coli that is symbiotic in our colon is absorbed by us. And so that's an example of a symbiotic relationship between humans and bacteria. We have all of this E. coli in our colon, and it is providing us with extra vitamin K, and we are providing it with some food and a nice, warm, protected place to live. You may find it interesting that, of course, that you may know this, you may have noticed this, but newborn babies, their excrement doesn't smell bad. Their excrement does not stink. And the reason for that is because they have not yet developed any bacteria. They haven't had any bacteria occupying their intestine yet. Where do they get the E. coli to put in their intestine? It's gotten in through food. So when a baby is nursing, they're just drinking milk or baby formula. They're not eating solid food. The solid food doesn't have any E. coli. They're not eating solid food that has E. coli in it, so their excrement doesn't smell bad. As soon as they start eating the baby food, the applesauce and all that stuff out of the jar, then their excrement will start to stink, because no matter what you do, you can't get rid of all the E. coli from that food. So there are little bits of E. coli that make it into your intestine when you start eating solid food. And the smell, the bad smell of human excrement and animal excrement, is actually caused by the bacteria. That horrible smell that you're smelling is not human excrement. That horrible smell you're smelling is actually E. coli. All right, so the remaining parts of the digestive tract, the large intestine, we start with the cecum that has the appendix attached to it. Then we have the ascending colon, the transverse colon, the descending colon, the sigmoid colon, the rectum, and the anus. Right, the cecum, the ascending colon, the transverse colon, the descending colon, the sigmoid colon, the rectum, and the anus. As I said, the colon contains symbiotic bacteria like E. coli that produces a lot of extra vitamin K that we get some of that, we absorb it. The E. coli is part of what's known as the natural flora of the intestine, also known as the microbiota of the intestine. So if you take Biology 205, we'll talk about the natural collection of bacteria that is found in the human intestine. And they're actually good for us. The fact that we have bacteria living in our intestine is not bad. We live in this symbiotic relationship. And they are what are called, we can sometimes refer to them as the good bugs of the human intestinal microbiota, because they don't do it, not only do they not do us any harm, but they actually provide us with good things. And more importantly, because they are there, they prevent bad guys from getting in. So there are a lot of bad bacteria that could get into your intestine and make you sick, but they can't because the good guys are already living there. Right? So examples of good guy bacteria are E. coli and several different species of lactobacillus, Lactobacillus acidophilus, for instance. Those are good bacteria that are part of the normal gut microbiome, as it's called, the gut microbiome, or the gut flora, the natural flora of the human intestine. As I said, babies have excrement that doesn't smell bad because they have not yet developed a gut microbiota. They haven't swallowed any bacteria, and those bacteria have not moved into the baby's gut yet, so the excrement doesn't smell bad until they start eating solid food. Okay, now one thing that you might be interested in, which is that antibiotics are chemicals that kill bacteria, specifically bacteria. And so if you get a bacterial infection, you get a streptococcal infection or something like that, they will give you antibiotics in the hospital. And those antibiotics will kill the bad guy bacteria that's in your blood or in some other part of your body. But the antibiotics also have the unintended consequence of killing the good bacteria that are in your intestine. And if you kill off the good guys that are in your intestine, the intestine is temporarily left unoccupied, and sometimes that allows bad guy bacteria to move in, gives them the opportunity to move in. Example, Clostridium difficile. Right, so C. difficile, Clostridium difficile, is a bacteria that lives in the soil, but food is made sometimes from vegetables. Vegetables are grown in the dirt. If you don't wash the vegetables properly, or if some of the C. difficile will get into the food you eat. And if the intestines are unoccupied as these C. difficile pass through your intestine, they'll say, "Hey, great, unoccupied prime real estate. Let's put up shop here." Right? And so then you get a C. difficile infection, which causes blisters to form on the inside of your intestine and causes you to have bloody diarrhea, often referred to as a nosocomial infection because you caught it when you were in the hospital as a result of a course of antibiotics. So normally these days, when you don't necessarily have to catch it in the hospital. It's a common thing that happens when you've had a course of antibiotics, even if you're at home doing the course of antibiotics. So these days, it is common practice that whenever you kill the symbiotic bugs, the symbiotic bacteria in somebody's intestine with antibiotics, you also prescribe them a course of probiotics. So the probiotics are the good guy bacteria. You put them back into your intestine before the bad guys have a chance to move in. I'm going to show you a slightly disturbing image of a C. difficile infection, which is what happens if you don't put the good guys back in your intestine. This is a normal colon, a normal healthy colon. Now I'm going to show you what happens if you have C. difficile there. So this intestine is a nice healthy colon that has a lot of E. coli growing on the walls. You can't see them because they're too small. But notice the walls are very shiny and glistening. That's the mucosa, that's the mucus. And then within that mucus, we have a lot of E. coli growing. And then as food comes down the colon, we also have E. coli mixed in with the food, the excrement, as it passes through. Now, if you have a course of antibiotics and you kill off all the good guy bacteria and C. difficile moves in your intestine, your colon will look something like this. This is called pseudomembranous colitis, where the colitis is an inflamed colon. So the colon is inflamed because C. difficile moved in and it caused blistering. So these blisters are covered with kind of a blistery membrane, so then it's called pseudomembranous colitis or C. difficile infection. Therefore, whenever we give somebody a course of antibiotics, penicillin, amoxicillin, and so on. If you're going to do that to somebody, then you have to go the extra step and give them probiotics. Right, so if you give somebody antibiotics these days, you're supposed to also put them on a course of probiotics to replace the good guy bacteria in the intestine. All right, we are continuing to talk about the hepatic portal system now. So the nutrients are absorbed from the small intestine through a network of veins called the hepatic portal system. They converge into a single large vein called the hepatic portal vein, and then go into the liver where they are detoxified. And then after having been detoxified, they are sent to the inferior vena cava through the hepatic vein. Hepatic portal vein, hepatic portal system. So again, don't confuse hepatic vein with the hepatic portal vein. They are two different things. Okay, so the liver creates bile, which is formed by the breakdown of red blood cells by the spleen. We talked about that when we discussed the blood. And then the bile salts are sent to the gallbladder where they're stored and then concentrated by absorption of water out of the gallbladder so that you kind of concentrate the bile in the gallbladder. And then from there it's sent to the duodenum through the common bile duct. The purpose is to emulsify fats, which makes it easier for the lipases to break them down. All right, you can see the gallbladder at the bottom of the liver and the pancreas located just behind the stomach. Okay, so the bile contains bilirubin, which is a yellowish product that results from the breakdown of hemoglobin. Right, so the greenish yellow color, the yellowish green color, is caused largely by bilirubin. Now, inside the liver, you know already that organs that have more than one piece, like if you have two pieces, the individual pieces of that organ are often referred to as lobes. And the liver is made out of a whole bunch of little pieces that we call lobules. And each one of these lobules is a filtering unit that contains its own vein and its own filtering cells, its own hepatic cells. So the liver, a healthy liver, is made up of a whole bunch of millions and millions of lobules. All right, I promised you that on the final exam I might ask a question, and I often do, is say, tell me 10 things that the liver does for 1 point each, and then you've got a 10-point question out of that. So let's just look at some of all the amazing things that the liver does. Okay, beginning with it recycles the hemoglobin, right? So it breaks down the heme group into bilirubin. It stores the iron and keeps that as well. So this is the hemoglobin breakdown into bilirubin is caused by the breakdown of erythrocytes by the spleen. The liver also stores several of the vitamins, including vitamin A, B12, D, E, and K. It creates the bile, which is used to emulsify fats during the digestive system. It stores spare glucose in the form of glycogen in case we have to fast for a while because we can't find any food. The liver will convert the glycogen back into glucose and release it into the blood. The liver also detoxifies all the nutrients that were absorbed by the small intestine. The liver converts ammonia into urea. That is part of the urea cycle, right? And that the urea is then filtered out of the blood by the kidneys, the renal system. When we discussed the immune system, when we discussed the lymphatic system, we talked about the fact that the liver creates the complement proteins rather. So when antibodies coat for an invader, that's called opsonization. And then the antibodies will bind to a bunch of proteins called complement proteins that are created by the liver, and those complement proteins will break holes in the invading bacteria or the invading viruses and kill them that way. So creation of complement is another nice thing the liver does. The liver also creates the serum albumin that regulates blood pressure. The liver also creates a lot of cholesterol that is used to put into all of the cell membranes, the plasma membranes, to make them more rigid. And the liver also creates the anticoagulant protein heparin, which prevents blood clots from forming inside blood vessels as long as the blood vessels are nice and intact. Okay, so the liver recycles hemoglobin, it stores vitamins, it creates bile, and it stores glycogen. Those are 5 things that it does. It also detoxifies the nutrients from the small intestine. It converts ammonia to urea, part of what's known as the urea cycle. Takes place in the mitochondria of hepatocytes. Creates cholesterol, creates the anticoagulant protein called heparin. It's not really a protein, it's a chemical. It creates serum albumin, and it creates the complement proteins, which are part of the immune system. So that was a whole bunch of really great things that the liver does. Okay, let's take a closer look at the pancreas. The pancreas produces digestive enzymes, carbohydrases, lipases, nucleases, and so on, proteinases, which are a class of proteases. And it also produces bicarbonate, and it produces all of these things in a type of cell called acinar cells. Now, the reason why we call them acinar cells is because the pancreas also contains other cells that produce hormones, insulin and glucagon, which we'll discuss when we discuss the endocrine system. Right, so the pancreas is an interesting organ. It's classified, it's included in 2 of the organ systems. It's included in the digestive system as well as the endocrine system. So today in this lecture, we're talking about the digestive system functions of the pancreas. And the digestive system functions of the pancreas are carried out by acinar cells. The acinar cells are mixed together in the same organ with cells that produce insulin and glucagon, which are part of the endocrine system, and we'll talk about that later. Okay, so the acinar cells are organized into little circular units that are called acini. The acini kind of line the pancreatic duct so that they dump their contents into the pancreatic duct, which then dumps them into the duodenum. So there is the pancreas behind the stomach. Here you can see the acinar cells forming acini, which line the pancreatic duct and lead into the duodenum. And as I said, the pancreas also produces insulin and glucagon, which we'll talk about later when we get to the endocrine system. Okay, so I mentioned the fact that the intestines are held in place by a serous membrane called the peritoneum. Like all serous membranes, it has a visceral layer and a parietal layer. The peritoneum is actually divided up into 5 individual folds. It's really just one membrane, but the folds are significant enough that they give them different names. And we're not going to learn all 5 names. We're only going to learn two of the names. And the 2 names that we're going to remember are the greater omentum, which covers the front part of the intestines and is used as a site for fat deposition, and the mesentery. Right, so the mesentery is a vertical band that's located just anterior to the spine, anterior to the lumbar vertebra, and it surrounds the intestines, but it also anchors the small intestines kind of holds them in place. Okay, so this is a transverse section through the human body, so you can see the greater omentum and the mesentery. This is a lateral view, right? So you can see the peritoneal cavity, you can see the small intestine running through it, and then you can see fat deposition in between the layers of the peritoneum. All right, so we've gone through the entire sequence of digestion. Food enters the mouth It forms a bolus when we chew it up and mix it with saliva. It's sent down to the stomach through the esophagus via peristalsis. In the stomach, it gets mixed with HCl and pepsin, also known as gastric juice. We rename the food chyme. The chyme then enters the duodenum through the pyloric sphincter, and then In the duodenum, the pancreas sends more digestive enzymes and bicarbonate. The liver adds bile salts, which help to emulsify fats. The food continues to break down as it transits through the small intestine, and the nutrients are absorbed by the hepatic portal system. And then the water is reclaimed in the large intestine, and then what's left goes out through the rectum. Now let's move on to some digestive system pathologies. Okay, I'll divide the pathologies into eating disorders. You've probably heard of anorexia and bulimia. Those are two eating disorders, more psychological issues than physical ones. Then we'll talk about some problems with the mouth, the teeth, and the gums. Gingivitis, for instance, you've probably heard of as being an infected gums. We'll talk about problems with the stomach and the sphincters. Acid reflux, for instance, is a problem with the cardiac sphincter. We'll talk about problems with the liver and the gallbladder. You've probably heard of gallstones. That's a common problem where these little things form in the gallbladder. Talk about some problems with the pancreas. Talk about problems with the large and small intestines. Okay, eating disorders. Anorexia is kind of a psychological disorder where people deliberately fast and they abstain from eating food because they have a distorted body image. They think of themselves as being too fat when in fact they're not. And so self-starvation, right? That is refrain from eating, starving yourself basically for not because you don't have access to food, but because you don't like your own body. Anorexia. Bulimia is a variation of anorexia where you eat everything you want, binge eating, and then you induce vomiting. You force yourself to vomit. That usually destroys your esophagus because it sends the hydrochloric acid, sends the gastric juice flying out through your esophagus, and so it burns your esophagus and maybe destroys it. It damages your teeth. If possible, it's anorexia. Anorexia is a very bad disorder, but bulimia is even worse for that. Bulimia is even harder on the body for that reason. Okay, tonsillitis, problems with the oral cavity. Tonsillitis, of course, is where the tonsils become inflamed, and usually that inflammation will go away, but sometimes they just say, it's easier just to remove them. Right? So when I was a child, thank goodness this is not true anymore, but when I was a child, they used to encourage children to have their tonsils out before they became inflamed. So when I was a child, it was a routine that in the second or third or fourth grade, the kids would start to have their tonsils removed. They'd take a day or two off class and they'd come back and they'd say, look, I had my tonsils out, and they'd open up their mouth and you can't see any tonsils anymore. But really, there's no need to remove them unless they're inflamed, and maybe they never will be. So in my case, for instance, my parents just didn't bother to have my tonsils out, even though there was a lot of peer pressure that all the other kids were having their tonsils out. They never bothered to have my tonsils out, and I have never had inflamed tonsillitis or anything like that. So in my case, that would have been unnecessary surgery. Okay, gingivitis, of course, is inflammation of the gums. That's usually when bad bacteria get into the plaque that's kind of stuck. If you don't do a good job brushing your teeth, you get bad bacteria kind of stuck up between the layer where your gums meet your teeth, causing a problem. Okay, so tonsillitis, you can see the inflamed tonsils versus regular tonsils. Gingivitis, we have inflamed gingiva. The technical name for the gums is the gingiva. So gingivitis, of course, is inflamed gingiva. Okay, so remember that we have the bottom part of the stomach is called the pylorus. If you have a gastric ulcer, that's usually caused by your own stomach acid and your own pepsin burning a hole in the inner layer, the lining of your stomach. Okay, 20 years ago, people would have happily told you that you have ulcers because you're leading too stressful a lifestyle, and it is true that if you're under long-term stress, your stomach then tends to produce more acid, but that turns out not really to be the reason why the stomach acid is able to burn your stomach. the reason the stomach acid is able to burn your stomach is because you've got an infection of a type of bacteria called Helicobacter pylori. If you take Biology 205, we're going to discuss Helicobacter pylori endlessly, but you can get rid of Helicobacter pylori with antibiotics. So Helicobacter pylori is a bacteria that eats the mucus, eats away at some of the mucus lining of your stomach and gives the acid and digestive enzymes a chance to burn away at it. So if you have ulcers and has nothing to do with a stressful life cycle or a stressful lifestyle, they just give you some antibiotics now. They don't tell you to take a vacation, which is too bad. But in the old days, they used to say, "Oh, you have ulcers. You have too stressful a lifestyle. Just go on vacation for a while and relax." And unfortunately, they don't say that anymore. They just throw some pills at you and say, "Take these pills, these antibiotics, until the Helicobacter pylori is gone." All right, so as I said, the word gastro refers to the stomach. So gastritis is inflammation of the stomach. And then gastroenteritis specifically means inflammation of the stomach and the small intestine, and various reasons why it might be inflamed. Okay, this is a kind of an interesting pathology, a hiatal hernia. Okay, so a hernia is where you have a tube or something in the body bulges, it forms a weakened bulge. And the spot where the esophagus goes through the diaphragm is referred to as the esophageal hiatus. The word hiatus means a gap, right? So a hiatus is a gap. So the hole where the esophagus goes through the diaphragm is called the esophageal hiatus. And a hiatal hernia is where the stomach kind of pushes its way up through the diaphragm so that you have a little bit of the stomach up in the thorax. You have a little bit of the stomach up in the thoracic cavity. And if you have this great big stomach bulge that's forcing its way up through the diaphragm, including the cardiac sphincter, the cardiac sphincter has a hard time closing, and so you tend to have really chronic long-term acid reflux. So here's a hiatal hernia, right? So here's a normal stomach, normal esophagus making its way through the diaphragm. And then here we have a bit of the stomach that's pushed its way up above the diaphragm, and we call that a hiatal hernia. All right, my favorite stomach disease, because I have this to a certain extent. Right? So gastroesophageal reflux disease is commonly referred to as GERD, gastroesophageal reflux disease. Right? Also known as heartburn or acid stomach or acid reflux. Gastroesophageal reflux disease is the technical name for it, and that means you have heartburn. You drink this stuff. A quick and easy cure for it is to drink this stuff called Pepto-Bismol. Bismuth is a base compound that will neutralize it. So Pepto-Bismol is a brand name, but you can get bismuth, generic bismuth, so it's not sold under a brand name for cheaper. That's the quickest way to cure acid reflux, gastroesophageal reflux disease. That's the quickest way to cure it, but that's a short-term cure. The long-term cure is to not do things that abuse the cardiac sphincter. Okay, so acid reflux or heartburn or pyrosis are all referring to the GERD, gastroesophageal reflux disease. It usually happens because the cardiac sphincter has become stretched and doesn't close properly, allowing the hydrochloric acid to reflux up into the esophagus. Typical causes for the cardiac sphincter to become stretched are chronic overeating. If you often eat too much, it's very hard to stop eating if you have a nice meal in front of you. But if you chronically overeat, then the stomach fills up so much that there's no room for the food to expand any further because the stomach can't expand any further. There's no room for the food to go, so it ends up pushing its way up through the cardiac sphincter into the esophagus, and that has the effect of stretching the esophagus. Stretching the cardiac sphincter, rather. And then if you eat before going to sleep, you're abusing the cardiac sphincter as well, because it's easier for the cardiac sphincter to keep the acid in your stomach if you're vertical and the esophagus is above the stomach. Excuse me. If you're horizontal, the stomach is at the same level as the esophagus, and it's easier for the acid to spill out of the stomach. Now that wouldn't necessarily be a problem if it's been a long time since you ate and your stomach is no longer producing the acid because of the somatostatin. But if you ate late, if you ate a midnight snack and then went to sleep and laid down, that's the worst thing you could do, because your stomach is busy trying to digest all this protein. It's producing a lot of hydrochloric acid, and meantime the esophagus is on the same level, so the esophageal sphincter has a hard time, or sorry, the cardiac sphincter has a hard time keeping the acid in the stomach under those conditions, right? So if you want to avoid gastroesophageal reflux disease, don't overeat and don't eat before lying down. I have to admit that I do both of those things quite often, so I'm a bad example, and I get punished for that because I have bad acid reflux a couple nights every week. You know, it stops me from sleeping properly, and I have to take some antacid like that. And I say, oh, I'll never eat a midnight snack like that again. I've learned my lesson, and then the next night I'm doing the same thing. All right, so that's gastroesophageal reflux disease. All right, you'll forgive me if I trip over my tongue when I pronounce some of these, but cholecystitis is inflammation of the gallbladder, right? And we can tell because there's referred pain sometimes in the neck, the upper right-hand side of the neck. Cholelithiasis is the formation of gallstones. So typically we mentioned the fact that the liver produces bile and then puts it in the gallbladder, and the gallbladder concentrates it by removing water. If the gallbladder removes too much of the water, the bile salt will actually precipitate. It will form a solid, and we call those, common name for those little solid green balls is gallstones. The technical name is calculi. So if the gallbladder produces too much bile, or if the liver produces too much bile, or if the gallbladder concentrates it too much, the salt will crystallize, and then you have calculi. Or if you have too much cholesterol in your diet sometimes, and too much cholesterol in the blood, you can also have calculi. And you have a sore gallbladder because you have referred pain as well. We discussed that. Right. And then finally, jaundice is where the bile that contains the bilirubin in the liver is not working properly. So the bilirubin doesn't all end up in the gallbladder. Some of it ends up getting backed up into the blood. And this, if you have bilirubin in your blood, it ends up in your skin and in your eyes. The whites of your eyes are referred to as the conjunctiva. And so then you get, you have this yellow tinge to your skin. Your skin appears to be yellow, and so do the whites of your eyes. That's called jaundice. All right, here we see the formation of calculi in the gallbladder. And I'm going to show you a disturbing image of a gallbladder with calculi in it. All right, so here you have a gallbladder with calculi that have been formed. This is quite bad. So typically the transit of the calculi down the bile duct is painful. So you get a little bit of pain from gallstones. Not nearly as much pain as you get from kidney stones, but gallstones, you can get pain from the calculi going down the bile duct. One way to cure that is through ultrasound. So they put you in a bath of water, and then they point an ultrasound gun at your gallbladder and send bursts of high frequency sound at it to break these calculi up into smaller pieces so they don't cause pain as they're going down through the gallbladder. And then once in a while, as a last resort, somebody's will have their gallbladder removed, and then they'll just connect the liver directly to the intestine. All right, now the liver can sometimes break down, and if the hepatocytes that form the liver die, and the lobules that the hepatocytes were forming die, and then they get replaced with scar tissue, we call that cirrhosis of the liver. Right? So the liver is the same size as it was, but it only contains half as many hepatocytes, and the rest of the volume is taken up by scar tissue, which does not do the same job of filtering things the way the liver does, the way the hepatocytes do. Right? So that is a bad thing, because then your liver is unable to filter the ammonia and other bad things out of your blood, is not able to convert it to urea, it's not able to break down peroxide and other things, and that eventually can kill you. So there are things you can do about that, but the causes are generally alcoholism. Right? So if you drink too much alcohol, whenever you drink alcohol in general, the liver actually works to detoxify it. Somebody who's a moderate drinker or a heavy drinker will start out by having an enlarged liver. So one of the tests that a good family physician should do is they should check to see how big your liver is by palpating it. You know, they thump around, they do auscultation, they thump around the area, thump around the right side of your thorax, just under the ribs, to see how big your liver is. And then they can tell how big the liver is. If the liver is enlarged, hepatomegaly, if the liver is enlarged, they'll ask you if you drink alcohol. And if you say no, then they say, oh, well, there's some other reason for this enlarged liver. If you drink a lot of alcohol, that could be the reason. They might advise you to cut back, because if you keep on doing this, the first sign of alcoholism in your youth is that your liver becomes very big, because it becomes efficient, because it's having to work so hard to detoxify all this alcohol. But it can't keep that up forever, and eventually the hepatocytes begin to die, and you end up with cirrhosis of the liver, which will eventually kill you. So chronic alcoholism is one of the main causes of liver failure, cirrhosis of the liver. But there are viruses that can kill the hepatocytes as well, such as hepatitis A, hepatitis B, and hepatitis C. If you take Biology 205, we'll teach you about all three of those viruses. Hepatitis A is a virus that does moderate damage to the liver. You usually get over it. Hepatitis B and C are much more serious. There are vaccines that prevent you from catching hepatitis A and B. If you're a hospital worker, if you work in a hospital, the main way that hepatitis B is usually transmitted is through IV needles. And so if you have a patient who has hepatitis B or C, and you accidentally pull out their IV needle and it ends up poking you, then you'll get hepatitis B. So as a precaution, most people who work in hospitals, phlebotomists and nurses and doctors, have to, if you work in a hospital, there's a rule that you have to have the hepatitis B vaccination, so that if you accidentally poke yourself with a contaminated needle, you at least won't get hepatitis B. I don't know if there's a vaccine against hepatitis C yet, but the last time I checked, there was not. So it's still dangerous. You know, you're still playing Russian roulette when you work with needles in a hospital. All right, here on the left we have a normal liver, and on the right we have a cirrhotic liver. So you can see that this nice smooth liver tissue has been replaced by this bumpy scar tissue. Let's have a look inside it. And so here we have all of this scar tissue inside the cirrhotic liver. As I said, the causes are alcoholism and hepatitis. Hepatitis is usually spread by needles, IV drug use. There are a few people in Canada who, in the early 80s, before they were aware of this, they got hepatitis from blood transfusions. They've now corrected that problem, so it's safe to get blood from the hospital now, because they now check for that. Hepatitis A is often caused by fecal contamination of food. That means the person who makes your salad has hepatitis A. It's a mild case, so they don't realize they're sick. They go to the bathroom, they go to the washroom, and they get excrement on their fingers. They don't do a good job of washing their hands. They still have a little bit of their own excrement under their fingernails. That's a common problem. Then they prepare your food, and then you eat it, and then you have hepatitis A. If you're young and healthy, you may not even realize you have hepatitis A, and it's self-limiting, which means you get over it all by yourself. If you're older and infirm, it could become a problem. As I said, there is a vaccine against hepatitis A. It's an oral vaccine, so you can just take it orally, and then you don't have to worry about that. However, one of the things that restaurants should be doing is getting all of their staff to wear gloves, either latex gloves or nitrile gloves. Sometimes you see restaurants using those food preparation gloves that are made out of plastic. That's better than nothing, but it's not as good as latex gloves. But a lot of restaurants are reluctant to have their cooking staff put on latex gloves, because then if the clientele sees that the chef is wearing gloves, they'll get nervous and say, why is the chef wearing gloves? Does he have some kind of disease? And they say, oh, no, 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 no. But that is a precaution that restaurants should be taking. The food preparation people should be wearing latex gloves or nitrile gloves. All right, let's look at some pancreas pathologies. Right. Pancreatitis just means inflammation of the pancreas, often caused by either alcoholism or infection. Diabetes is a problem with the pancreas, but it doesn't affect the digestive functions. So we're going to talk about diabetes when we discuss the endocrine system. Okay, so enteritis, the word enteritis means inflammation of the small intestine. Gastroenteritis means inflammation of the stomach and the small intestine. And then, of course, ileitis refers to inflammation of the ileum, the latter part of the small intestine. Okay, so as we said, the main purpose of the large intestine is to recover water. So diarrhea is where you eat something bad and your intestines say, get this out of here. We don't care about losing water, just get it out of here fast. And diarrhea looks much, that is basically what food looks like when it's in the small intestine. Okay, so if the large intestine fails to recover all of the water, you will have diarrhea. If the large intestine takes too much of the water out, it'll cause constipation. And so that because it took too much of the water out in the stool, the excrement has a hard time getting out of the rectum because it's too hard. Now, this can often be caused, constipation can often be caused by a fatty diet, because there's a hormone that is produced when you eat fats. The hormone tells the intestine to slow down and move the food through the intestine more slowly, because lipids contain so much energy, on a kilogram per kilogram basis, lipids contain more energy than any other molecule. So from our primitive days of being a hunter-gatherer society, when we didn't get to eat very often, when you managed to eat a fat or a lipid or an oil, it was a real treat. And so you didn't want to digest that quickly, because you might waste some of the goodness, some of the food energy. And so when you ate fats, your intestine would produce this hormone that tells the smooth muscles to slow down with the peristalsis, just slow down and move the intestine through the food more slowly, because we want to get everything we can out of this. And if the food moves through the intestine more slowly, that means the colon has more time to suck the water out of it, and therefore that ends up being harder than it should be, and it's difficult to get rid of. All right, some other problems. Diverticulitis is a disease common in older people, where the connective tissue and the muscles in the intestines become weak, and they kind of develop hernias, kind of develop bulges. And the bulges are referred to as diverticulae, individual diverticula. And food can get trapped in the bulges, and it causes irritation, and it's very painful. So this is a common thing in older people. They have these stabbing pains in their abdomen as a result of diverticulitis. Colon cancer, of course, is a big problem. This is where some cells, because the cells on the inner side of the intestine are constantly being replaced, that's one of the cells that's dividing in the adult human being. Because those cells are constantly being replaced, they're subject, or they're more vulnerable to becoming cancerous. Basically, the more times a cell divides, the greater the odds are that it will become cancerous, because something happens to the DNA when it's being replicated to make a new copy of the cell. So any cells that are constantly being replaced are more in danger of becoming cancerous than cells that are not dividing all the time. So colon cancer is something that you might get. It usually starts out as a polyp. A polyp is a little tiny light bulb shaped bulge in the inside of the intestine. You can check for polyps by doing a colonostomy. Colonostomy is where you remove a section of the intestine. Colonoscopy is where you put a scope into the intestine. So colonoscopy is where you take a scope and you look inside the intestine to see if you can find any polyps. If you find polyps when they're small, then you can cut them out and you probably don't have to worry about colon cancer. If you catch them when they're large, they might have already metastasized into cancer, so that would be bad. So if you find colon cancer, you can actually take a section of the colon out and sew the other sections that are left together, right? Or you can just chop off the lower part of the large intestine and leave it chopped off and replace it with a bag, and that's called a colonostomy. All right, finally, hemorrhoids. Hemorrhoids are where you have varicose veins in the rectum. Then you have these swollen veins in the rectum that may actually come out. And remember, when the organs face the wrong way or fall out of the abdominal cavity, that's referred to as prolapse. Right? So if you have these swollen varicose veins actually coming out of the anus, as the excrement goes by, it hurts, and that is referred to as hemorrhoids. And you can remove the hemorrhoids surgically. If you don't, they're actually quite painful. All right, so this is just demonstrating diverticulitis. So diverticulitis, you have these bulges that kind of form on the weak spots that form in the intestines, and it's very painful as the food moves by. This is demonstrating colon cancer and polyps. So we have stage 0, stage 1, stage 2, stage 3, and stage 4 is the largest of the polyps. That's usually the worst, of course. If you have them when they're in stage 1 or something, you're doing a colonoscopy, you're looking. If you see a stage 1 polyp in there, you can usually just chop it out with a little caliper that you insert in with a wire, so you don't even have to do any surgery. And then you can stop the cancer there. This is a giant inflatable colon that was made by the Canadian Colon Cancer Society, and it's touring around the country. And if you ever get a chance to see it, if it ever comes to Vancouver, or if it ever comes to your town, wherever you're living, you should have a look inside. And one of the things that it's showing inside here, so this is a giant inflatable colon that you can walk around inside and look. And these things that are on the walls are actually polyps that it's showing you. All right, so if you have a legitimate colon cancer, you can actually chop out a piece of the colon, because the different sections of the colon are supplied by different sets of blood vessels. So it is possible to actually chop out a piece of the colon and sew the other parts back together. Now, if you chop off the bottom part of the colon and then just leave it off, and then you attach a bag, you hang a bag outside of the body to collect the excrement. That's called a colonostomy. And so there are people in the world that are walking around with a bag full of excrement under their clothing, because unfortunately they have had colon cancer and they've had to have the bottom part of their intestine removed. Okay, but if a section of the intestine has cancer and you're able to remove it, you can actually chop the middle section out and sew the other two pieces back together. So that's called a bowel resection. And if you do it like this, that's an end-to-end bowel resection. Sometimes you sew it together like that, and it's referred to as a side-to-end bowel resection. And sometimes you sew the sides together side by side, and that, of course, is a side-to-side bowel resection. Okay, so here's an unfortunate person who, for whatever reason, had to have their large intestine completely disconnected. So you can see this little pink thing here that's leading into the bag, the colonostomy bag, or also known as a colonostomy pouch. That little thing there is the end of the descending colon, which is emptying the excrement directly into the bag. So that means that every few hours, these people have to go to the washroom and empty the bag into the toilet. It's not very sanitary, but if you have colon cancer or a few other things, you may have no choice. All right, hemorrhoids, of course, is where you have varicose veins that are actually swollen and coming out of the anus. And so when excrement goes by, it's painful, and you can remove those with surgery. All right, and appendicitis is where the appendix becomes inflamed. It's a vestigial immune organ. You remove it, that's an appendectomy. So there's the appendix sitting on the bottom of the cecum. If it becomes inflamed or if it starts to leak, that's called a perforated appendix. That's no good if it starts to leak, because if it leaks, then you have excrement that's leaking into your abdomen, and then you can get septic poisoning because that excrement is filled with bacteria. So you have to either repair the appendix or take it out and sew the hole up so that you don't have any leakage. So here we have a normal appendix versus an inflamed appendix. You can see again here that the appendix is hanging off the bottom of the cecum. Okay, now where would the appendix be if you were a surgeon who was looking for it? Let's say you're a surgeon and you have a patient laying on the table, and your job is to remove their inflamed appendix. You're going to do an appendectomy. How are you going to find where the appendix is? You're just not going to cut. You don't want to cut a bigger hole than you have to. So what you want to do is to cut a hole that's directly above the appendix. If you have the patient laying down in front of you, you want to cut a hole that's directly above the appendix. So how do you find the appendix? The appendix happens to be located at McBurney's point. So I mentioned that there are a few anatomical landmarks that we're going to learn the names of in this course. McBurney's point is located 2/3 of the distance. 2/3, if you draw a line between the navel and the right anterior superior iliac spine, remember that the anterior superior iliac spine is the kind of the crest of the pelvis, and you can feel it with your thumb. You can feel it no matter how much adipose tissue you may have there. Right. So if you have a big stomach, you can still feel where the right anterior superior iliac spine is. And so that's, if you draw a line between the navel, the belly button, and the right anterior superior iliac spine, 2/3 of the distance from the navel to the spine is McBurney's point. And if you cut in that area, you'll be right above the appendix. Now, if you push on McBurney's point, you will cause referred pain in the epigastric region. Epigastric, of course, means the area right above your stomach, sort of below the xiphoid process of the sternum. So you kind of feel pain in your solar plexus. If you push on McBurney's point, that's referred pain that's coming from the appendix, and that's referred to as Aaron's sign. Right? Aaron's sign, very important. Remember what Aaron's sign is. Remember where you can find McBurney's point. Right, so there's the anterior superior iliac spine. There's the navel. We draw an imaginary line between those 2 points, and then you go 2/3 of the way from the navel. You cut right there. That is McBurney's point, and then underneath there you'll find the appendix. Just showing McBurney's point again. There's the epigastric region. So if you push on McBurney's point around there and you feel pain here, that's an indication that you have an appendicitis. You have you have an inflamed appendix. All right, to summarize, we talked about the structures of the alimentary canal, the mouth, the pharynx, and so on. It's a long continuous tube. We know which order the different parts of the intestine are in, right? So we have the stomach, which leads to the duodenum, then the jejunum, then the ileum, then the cecum, then the ascending colon, the transverse colon, the descending colon, the sigmoidal colon, the rectum, and the anus. That's the alimentary canal. We have the accessory organs, the liver, the gallbladder, the pancreas, and the salivary glands that put things into the alimentary canal as the food that's going past is being digested. We discussed a number of different pathologies of the digestive system, and we discussed where McBurney's point is, why it's important to know where it is, and what Aaron's sign is. Okay, I believe our next lecture is on the I don't remember. You'll figure it out. It's either the respiratory system or the lymphatic system, I think. Or the respiratory system or the endocrine system, I believe. All right, I'll see you there.